extrinsic protein. Extrinsic protein. So guys, now you can understand the glucose. Glucose need to enter inside the cell. If it is going inside the cell, guys, this need to cross the cell membrane. Guys, now please answer me, via which layer this glucose will cross? Via protein layer or via lipid layer? Because suppose this is the cell membrane, this cell membrane is having only the two areas. The number one areas which is made up of the protein, and this is for the lipid, suppose. So this glucose will cross the cell membrane via the protein layer or via the lipid layer. Now please answer me. Yes, guys. Yes. Very good, guys. Very good. Well done. Why protein layer? Why protein layer? Because glucose is water soluble. चीनी भैया आराम से पानी में मिक्स हो जाती है. चिकन जी बना लेते हैं, पी लेते हैं फटाक से. चीनी मिक्स हो रही है वाटर के अंदर. चीनी, sugar is not going to mix into the oil. You always can mix this sugar only in the water. So this glucose is going inside of the cell. It will cross via the protein layer. Right? Very good, guys. Very good. Now, suppose you are consuming what? Your vitamin E. Your vitamin E, if you are consuming, via which lipid layer it will cross? Yes, please answer me. Vitamin E. Via protein layer, lipid layer. Yes, guys. Yes. Everybody perfect, guys. Well done. बहुत बढ़िया. बस ऐसे आंसर आने चाहिए बेटा. Now definitely आप एनडी को दिखा देंगे कि yes, you can do this. There is no any exam in this world which you can't pass. You can pass any exam in this world. और एनडी चाहे कैसा भी पेपर बना दे, आप एनडी को फोड़ के आने वाले. आप पेपर को 100% फोड़ के आने वाले. Guys, I give you the guarantee for that. 100%, right? So this is how basically you should answer. Very good. So this integral protein, this is basically passed through the entire thickness. This passed through the entire thickness. This passed through the entire thickness, and they tightly bound with the membrane. They tightly bound with the membrane. Peripheral protein present on peripheral side, and they loosely bound with the membrane. They loosely bound with the membrane. Now talking about the example of this integral protein. So integral protein example is number 1, cell adhesion protein. Cell adhesion protein. Then few receptors. The most common receptor which is present on the cell membrane is GPCR. GPCR stands for G protein coupled receptor. GPCR stands for G protein coupled receptor. Then few pumps like sodium, potassium, ATPase pump, sodium, potassium, ATPase pump, channel via chloride channel, which is regulated by CFTR gene. Which is regulated by CFTR gene, and this CFTR stands for cystic fibrosis transmembranous regulator. If this gene defect, this can lead to a condition called as cystic fibrosis. So defect can lead to what? Cystic fibrosis. So this is pumps, that is the sodium, potassium, ATPase pump and the chloride channel. And this was the question which asked on the 6th of July FMG exam 2024. That CFTR gene regulates which ion channel? That is our chloride ion channel. Right? Talking about the peripheral protein. The peripheral protein is example is supportive proteins. Supportive proteins, they are present like the cytoskeleton. Like number 1 is dystrophin. This is present mainly in skeletal muscle. If it is get defected, this defect can lead to DMD, stands for Duchenne muscular dystrophy. This is the problem of dystrophin protein. Ankyrin and spectrin, this is present in RBC. If this is going to be defect, if ankyrin will be defect, what will be happen? This can lead to spherocytosis. So this biconcave shape of this RBC not able to be maintained, and if biconcave shape is not able to be maintained, this RBC will change its shape, and after that this is going to be stuck in the smaller side of pipeline and it will be degrade or destroyed, and this can lead to the less number of RBC called as anemia. So this is called as lower. This is due to the breakdown of the RBC, so called as hemolytic anemia. Right? Sure, Vishik, I will mark that. Right now this GPCR, guys, please write it down. Few things about this GPCR as well. This GPCR can be two types. This is having two pathways. That is number 1, adenine cyclase. This is adenyl cyclase, and another one is phospholipase cyclase. Another one is phospholipase cyclase. This adenyl cyclase will activate the CAMP, and this CAMP actually activates the pKa, and this pKa stands for phosphokinase A. Right? And this phospholipase cyclase, this can be activated two things. It can be activated the IP3 and it can be activated the DAG, because these two also secondary messenger. So this IP3, when it will be activated, it will release the calcium, and if DAG will be activated, it can activate or it can release the PKC, and PKC stands for phosphokinase C. So this is how this pathway is actually regulates. Right? Now So this is the story about the cytoplasm, or this is the story about the cell membrane. Now we are going to talk about the cytoplasm. Cytoplasm, it's 80% water, it's a clear fluid, and that's why it is also called as cytosol. And it contains the carbohydrate, lipid, electrolytes, everything. Talking about the nucleus, the third component of the cell, nucleus, it is the largest cellular organelle, and the diameter is 20 to 22 micron. So if the question comes, what is the largest cellular organ? That we call as nucleus. What is the largest organ of the body? Now please answer me guys. What is the largest organ of the body? Yes? Please answer, what is the largest organ of the body? Very good. The largest organ of the body is skin, not the liver. Liver is the second largest organ because the skin is also made up from the cell. Whatever is made from the cell, it makes the tissue, tissue makes the organ, and organ makes the system. So this is also the organ. So skin is the biggest organ of the body. Now talking about the membrane transport, how anything goes inside the cell, outside the cell, and that we are going to talk about the membrane transport. Whatever substance is moving across the cell membrane, it can cross the cell membrane by two mechanisms. Number one, either without energy or either with using of the energy. If any substance moving without using of any energy, that we call as passive movement. If it is moving via using of the energy, we call as active movement. So active means with energy and passive means without energy. Right? Now, so passive means no energy is required, and this from this table, guys, 100%, 1% will come in your FMG exam in the December. So passive means this is no energy required, no energy required, and active means energy required, energy required. Passive means no energy required means suppose you are standing on the top of the hill, and if you are throwing any stone, it will go where? Downside. So it is coming from higher side to low side, and this does not require any energy. So this is also called as coming from up to down, so called as a downhill phenomenon. Right? So this is passive means it is always from high concentration to low concentration movement, and this is also called as downhill phenomena. Downhill phenomena. Active will come from low concentration to high concentration, means now you are standing at the base of the hill, and now you are throwing the stone to the upside, required energy. So this is low to high, and it is also called as uphill phenomena. Uphill phenomena. So two type of membrane transport, active and passive. And now, both in this active and passive, there is four subtypes. How many subtypes? There is four subtypes there. So first of all, talking about in the passive, the number one subtype, that is the simple diffusion, then facilitated diffusion, non-ionic diffusion, and osmosis. Please revise with me. Simple diffusion, facilitated diffusion, non-ionic diffusion, osmosis. Simple diffusion, facilitated diffusion, non-ionic diffusion, osmosis. Simple diffusion, facilitated diffusion, non-ionic diffusion, and osmosis. Talking about in active, again there is four. Number one is primary active, number two secondary active, then exocytosis, then endocytosis. So number one is primary active, secondary active, exocytosis, endocytosis. Primary active, secondary active, exocytosis, endocytosis. Primary active, secondary active, exocytosis, endocytosis. Now close your eyes and now revise with me. There is a passive, there is an active. In passive, number one, that is your simple diffusion, facilitated diffusion, non-ionic diffusion, osmosis. Simple diffusion, facilitated diffusion, non-ionic diffusion, osmosis. Talking about in active, primary active, secondary active, exocytosis, endocytosis. Primary active, secondary active, exocytosis, endocytosis. So I hope now you can easily remember that's 4 in passive and 4 in active. Now let's talking about the example. Example for the simple diffusion is all gases. Suppose oxygen is coming into the blood. Through this blood, it needs to enter into the cell. So this oxygen higher side at the blood vessel, and it is diffusing to the lower side. So going from high to low does not require any energy. So this is the passive diffusion. So example of the simple diffusion is all gases. All gases, and they depend upon a fixed law. They depend upon fixed law. In the respiratory, we are talking about what is the fixed law. Then talking about in the facilitated. Facilitated is, this is number one is glut, which is for the glucose, and number two is aquaporin. Aquaporin, which is for the water. Aquaporin for the water. Glut stands for glucose transporter. Later on, we'll discuss about what is the glucose transporter. Non-ionic diffusion. The three components are there for the non-ionic diffusion. Number one, that is ammonia in kidney. Ammonia in kidney. Then bile salts in terminal ileum. And number three is salicylate in stomach. Salicylate in stomach. Salicylate is present into the paracetamol or the aspirin. So basically this aspirin absorption happens in the stomach. This is the salicylate in stomach. So all these three examples, these are very, very important for our exam point of view. So please be remember these three examples for non-ionic diffusion. Ammonia in kidney, bile salts in terminal ileum, and salicylate in stomach. Okay, so three examples of non-ionic diffusion. Talking about osmosis, you know very well about the osmosis. Osmosis means if a high concentrated or high solute solution is present on a one bottle, and in another bottle there is a low solute concentrated water is there. Now what will happen if there is a membrane is present? What will happen? This higher amount of the solute, they will absorb the water. So what will happen? This water starts moving from low solute concentration to high solute concentration. This condition we call as osmosis. Okay, so this we call as osmosis. Now talking about that primary active. Talking about the example of primary active. The primary active example number one is sodium potassium ATPase pump. Sodium potassium ATPase pump. Example of secondary active, that is your SGLT. That is your SGLT. SGLT. SGLT stands for guys sodium glucose linked transporter. What we call this SGLT? Sodium glucose linked transporter. Sodium glucose linked transporter. So this is your SGLT. Guys, this SGLT is two types. That is SGLT1 and SGLT2. Remember where they are present. Location is very, very important. So this SGLT1 and SGLT2. SGLT1 is present. SGLT1 is present in intestine, and 2 is present in kidney. You can remember something like that. That this eye is looking like this eye. So eye for eye, SGLT1 in intestine, and how many kidneys are there? There are 2 kidneys is there. So SGLT2 present in kidney. Very, very important part. Another example, apart from this SGLT, another example of the secondary active transport is sodium iodide symbol. Guys, why we require this iodine? Iodine is required for the thyroid hormone formation, and this thyroid hormone formed at the level of thyroid gland. So this sodium helps for this iodine to enter into the cell, and this type of transport we call as secondary active transport. Guys, please remember that suppose this is your thyroid follicle. So this is having this type of parafollicular cells, and this is your blood vessel. If suppose this iodine is coming, this iodine first of all need to enter via this cell, this membrane. This need to enter via first of all this membrane, right? So at this area, the channel which is present over here, this is we call as sodium iodide symbol. Sodium iodide symbol. But then this iodine need to again cross this membrane so that it can reach into center of the cell. So at this area again it is having the channel, and this channel name is pendrin. This channel name is pendrin, right? So then this iodine will enter via this pendrin, and then it will enter like this. So this is how this is actually presents at this area. So sodium iodide symbol and the pendrin, two channels are there for the iodine, so that it can produce the thyroid hormone. Now, what will happen, guys? If suppose this pendrin get defect. If this pendrin get defect, what will happen? This iodine cannot enter into the cell, and what will happen? No thyroid will be produced. This condition can lead to goiter. This condition can lead to goiter, right? This can lead to goiter. Now, this can also produce SNHL. This can also produce SNHL. In the end, you will study that sensory neural hearing loss, right? Sensory neural hearing loss. This is SNHL, right? So this is SNHL and goiter. This is the two areas. So this is your colloid. This we call as colloids, and this is we call as follicular area. This is the colloids area, and this is the follicular area. So this is how your thyroid gland will accept this iodine, right? Now the third component of this active, that is our exocytosis. Exocytosis. This exocytosis, first of all, it depends upon the calcium MCQ. So which ion dependent this exocytosis? Calcium dependent, and it also required a protein, called as snare protein. Very important part. Snare protein is present. What is the meaning of exocytosis? Meaning of exocytosis is that, guys, suppose this is your cell, right? This is your cell. This is your nucleus. This is your blood vessel. Now, what is the meaning of exocytosis? Exocytosis means any substance which is present inside of the cell that much, and it want to get out from the cell. If it want to get out from the cell, suppose this cell is having a channel over here. Now, guys, what do you think that if this product want to get out from the cell, it can move out by this channel? No, because this channel is very small. It cannot be accommodated into this channel. So that how this product can get out from the cell, and any product which is bigger in size and which can want to get out from the cell, this we call as exocytosis. What we call this? Exocytosis. Now the question is coming, sir, first of all, you just tell me or just let us know, sir, how this product enter into the cell. Guys, this product doesn't enter into the cell, actually it's formed into the cell. And right now, suppose you have a beta cell of the pancreas. They basically secrete the insulin. They synthesize the insulin and they secrete the insulin, which helps for the glucose absorption. Right now, suppose you are not eating any food and continuously your beta cell is producing the insulin, and this insulin is not coming out from the cell or from the gland, then it will not enter into the blood, because it will get out only when the boss will send the command. And when the boss will send the command, when you will consume the food. So right now, this insulin is making inside of the cell, and this cell keeping this insulin into the bag, and the size of this bag is going to be very big and thick. Now this big bag, now it cannot get out via any channel. So how this product can get out from this area? Or getting out this product, what this cell will do? Basically, this cell will break this cell membrane, and then it will release outside. So first of all, what will happen? First of all, the calcium will come inside whenever the message will come. This calcium activates a protein which is attached over here, the snare protein. This will attach this product over here. The lysis of the membrane will be happen, and the product will release out, and again the new membrane formation will be happens. So if any product is getting out via the cell, via breaking the cell membrane, this process we call as exocytosis. What we call this is exocytosis. So this is the exocytosis. Now another term, endocytosis. Endocytosis means any bigger substance which is present outside, it cannot again accommodate through this channel. So how this product can be get inside of this area? So now the question comes, inside there was the hormone, but what is outside? Outside it can be your bacteria, right? So this bacteria can't get entry via any channel. So what will happen? This cell will make a coat with help of a protein, and this protein is actually called as the clathrin protein and caveolin protein. And this is how this cell membrane will be break up. This bacteria will enter, lysosome will attack, and this is how this will keep this area neat and clean, right? So this is called as endocytosis. Means anything is going inside of the cell. So use the word endocytosis. So please write it down. This exocytosis, calcium dependent, insulin release from beta cell of pancreas. This another example is NTS. NTS means it's neurotransmitter. What is the meaning of neurotransmitter? Guys, we know inside our body we are having the connection between the boss and the organ, and this connection will be done by the nerve, and this nerve sometimes they attach with one nerve to another nerve via using of a synapse. This synapse is having a gap, and they will always talk, or they will always communicate via using of a chemical. This chemical is functioning like the transmitter signal, so that's why this chemical is called as neurotransmitter. And these neurotransmitter could be acetylcholine, substance P, norepinephrine, epinephrine, histamine. All of these are what? That is your neurotransmitter, right? Now the next part, that is your endocytosis. Endocytosis, it is a clathrin protein dependent, caveolin protein dependent. Clathrin protein, caveolin protein. There is a two process, pinocytosis, phagocytosis, and this we call as the receptor mediated, pinocytosis and phagocytosis. So pinocytosis is drinking of the cell, phagocytosis means eating of the cell. Now let's talk about this two things. Number one is what happens that whenever whenever we consume any food particles, right guys? Suppose what will happen? You will consume it from where? You will consume it from the mouth. So suppose you are eating the gulab jamun, right? So this is your gulab jamun. What will happen? This gulab jamun will enter from this area and it will go over here and it will digest, and ultimately this gulab jamun will release what? This will release the glucose, right? Now this glucose needs to be absorbed. So this intestine is actually made something like this. This is your intestine. Right? I told you inside the body everything is made up of the cell. So this intestine is also made up of the cell. So suppose this is the intestinal cell. And this intestinal cell is actually called as enterocyte. So I'm not making all these cells, right? All these cells. I'm not making all these cells. So these are called as enterocyte. Now this middle portion of this cell, intestine, this middle portion we call as lumen.